In the last video, we had a brief introduction to amino acids, and I mentioned a few little fun facts or just sort of things that you needed to know about amino acids when it came to discussing biochemistry. What we're going to do is we're going to continue our talk of amino acids, and we're going to get into the different things that you need to know about each specific amino acid. So, things that you should know for biochemistry about amino acids are their name, of course, their name, their structure, their three-letter abbreviation or code, their one-letter abbreviation, and just sort of important characteristics, other important characteristics that are specific to different amino acid. So I'll put here important characteristics or fun facts. Okay, so that's just a few things that we should know about each amino acid. And in this video in particular, I want to talk about the nonpolar amino acids. Okay, so of which I mentioned there were 20 amino acids in the last video. That's what I mentioned, and I talked about how, given the structure of an amino acid, the Fischer projection looks like this, with the carboxyl group up here. Oops, it's negatively charged. This is, of course, at a pH of 7, right, or a neutral pH. The amino group here on the left, protonated. Carboxyl group, I forgot to mention, is deprotonated. And of course here this R group, or the side chain. And this is what actually is different between each amino acid. These three things here, these three substituents, are all constant between all 20 amino acids, but there are 20 different R groups or side chains that makes each amino acid unique. So in this one we're going to talk about the nonpolar amino acids. Now, how many of them are there? How many nonpolar ones? Well, there are 20 total, but nonpolar there are 9 of them. Okay. So in this video, what I wanted to do essentially was to draw the 9 different nonpolar amino acids so you can know their names, their structures, their abbreviations, and important little things to keep in mind about each one. So let's just get right into it. So the first one I'm going to draw looks like this. Okay, and the R group is just an H. If you recall, that was glycine, right? So this amino acid here is glycine. Three-letter code for glycine is GLY, and the one-letter code is just G. Okay, so we need to know the name, the three-letter abbreviation, and the one-letter abbreviation. So if someone, maybe a professor or a teacher or a TA, whatever it is, asks you to draw glycine, they might not tell you they might not ask you just draw glycine. They might say draw the amino acid abbreviated as GLY, or draw the amino acid G. And as far as why we have these letter abbreviations, I mean, in science oftentimes things get really complicated, so shortening things down is helpful. So you need to be familiar with the idea that this structure goes with this name and these two different abbreviations. Okay, now there's something specific that I mentioned about glycine when we went over it in the last video, and I mentioned that it was not chiral, right? And the reason why is because it doesn't have 4 different substituents. Its R group is simply an H, and in order to be chiral, you have to have 4 different substituents on the carbon. So glycine is unique in that it can't be in the L form or D form. Oh, by the way, all the amino acids I'll be drawing will be drawn in the L form, right? So let me just note that here. So this is, of course, L form. And if you're unfamiliar as to what that is, I encourage you to look at the past video. Okay, so the first one is glycine. Okay. The next one, now actually henceforth what I'll be doing is, instead of drawing out the carboxyl group, the amino group, and the H, I want you to understand that I'm just going to not draw those groups just for simplicity's sake. Henceforth I'm just going to draw the R group just to save time. Okay, I don't want you to have to watch an hour-long video. So the next R group is simply just a methyl group. Now, this amino acid is called alanine. Its three-letter code is ALA, and its one-letter code is A. Often, you'll find that the amino acid three-letter abbreviation is just the first three letters of the actual name, and likewise with the one-letter abbreviations, it's often just the first letter. That isn't always the case, as we'll know, because there are some amino acids that sort of, their name starts with the same letter, so that can't be the case every time. Are there any particular fun facts about alanine that you should be familiar with? Well, not really. It just has that methyl group. Nothing too exciting about alanine. And again, keep in mind that the carboxyl group is still supposed to be drawn here, the H is supposed to be drawn here, and the amino group is still here. I'm just keeping it out of there, omitting it just for simplicity's sake. Okay. This next one I'm going to draw here is looks like this. So we have attached to the alpha carbon is a CH, and then there's a methyl group here and a methyl group here. Now this amino acid is called valine. Valine's three-letter code is VAL, and its one-letter code is V. Nothing too crazy about valine in terms of fun facts, but what I want you to notice is that you can remember Valine's structure by kind of remembering the idea that it looks kind of like a V, right? It's an upside down V here. So that can, just kind of keeping that in mind, can help you remember the structure of valine. Okay. So the next one I want to draw looks like this. Okay. So we have CH2, CH2, S, and CH3. This amino acid, its name is methionine. Methionine. Three-letter code is MET. One-letter code is M. Okay. Now, this one's a little bit different, and there are a few fun facts I want you to keep in mind about methionine. Now, methionine is you might recognize that name, and the reason why is if you took an earlier biology course, you might remember that when we were talking about translation, the start codon, right? The start codon in translation is AUG, right? Which codes for the first amino acid is methionine, right? So it is the first amino acid in many proteins. So when translation occurs, the first amino acid implemented is methionine. Okay, so that's a fun fact that you should keep in mind. Another fun fact about it is that it's one of two amino acids that are sulfur containing. So in the last video I mentioned AA, sort of my abbreviation for amino acid. Methionine is one of two amino acids that has a sulfur. Okay, so it's one of two amino acids containing sulfur. Again, so that's just another thing to keep in mind. Just a reminder, these ends here, they still look exactly like this, right? I'm just not drawing them to save time and probably space as well. Okay. So those are the fun facts I want you to keep in mind about methionine. So that's 4 of the 9 amino acids so far. The next one looks like this. Okay, now this one looks like this. Has a CH2 attached to a CH, and then that CH is attached to 2 different methyl groups. Now, this amino acid is called leucine. Leucine. Its 3-letter code is LEU. one-letter code is L. So far, these three-letter codes and one-letter codes have been pretty self-explanatory, and they pretty much are for the nonpolar amino acids. Leucine, if you notice, it looks pretty much like valine. The only difference, right? The only difference between them is that leucine has this CH2 before it actually splits off into this V shape here. So if you kind of keep in mind that valine and leucine have a very, very similar structure, obviously that can help you, right? That can help you remember these structures and whatnot. Okay, so that is leucine. The next one I'd like to draw looks like this. Okay, this next one looks like this. We have a CH here, and then we have a CH3, which is the methyl group, and then here we have CH2, and then off of that there's a CH3. Okay, so there's a CH here, much like in leucine and valine, and that CH has a methyl group, and then not just two different methyl groups, it has a methyl group and an ethyl group here. So leucine has the same number of carbons. Notice this has 1, 2, 3, 4 carbons, right? This has 1, 2, 3, 4 carbons in its side chain, and they're actually structural isomers. And so this amino acid here is called isoleucine. Okay. Now this is where the three-letter codes get a little bit trickier. It's not ISO, the three-letter code. It's actually ILE. So let me actually make that a little bit more specific. ILE. And its one-letter code is I. Okay, so in this case, this is one of those cases in which the three-letter code is not the first three letters of the name of the amino acid. So there is a fun fact that you should keep in mind about isoleucine. First of all, that it's a structural isomer of leucine. Okay. Of leucine. Excuse me. Another thing is that it's an amino acid that actually has two chiral centers. It has two chiral centers. What do I mean by that? What do I mean by that? Now, notice glycine doesn't have a chiral center, but all these other amino acids, their alpha carbon, right, has 4 different substituents. Now, isoleucine also has that alpha carbon, right, that is chiral, but now this carbon here also has 4 different attachments to it. This whole chain here with the amino group, the carboxyl group, and the H that are not drawn but are implied, as I mentioned before, the hydrogen and this methyl group and this ethyl group. So it has 4 different substituents on this carbon. So this has 2 different chiral centers in this amino acid. So that's also something to bear in mind when thinking about isoleucine. Okay. So let's make some more space. Now that's what is that? That's 1, 2, 3, 4, 5, 6 of the 9 nonpolar amino acids. Let me scroll down a little bit and draw these next few. These next 3, right? So this next one looks like this, and this one's a little bit weird. So I'm actually going to draw the what's it called? The carboxyl and amino groups. So I didn't draw them earlier, but now I'm going to draw them a little bit differently. So this one looks like this. That might freak you out right now, but these are just carbons here. So this is a CH2, this is a CH2, this is a CH2, and this here is the amino group. Now, what's crazy about this amino acid is that the R group actually comes back and connects to the amino group. So notice in the other amino acids, I drew the amino groups as NH3 pluses, right? So the N has 4 bonds to it, right? The bond to the alpha carbon and to these 3 different hydrogens. So that's 4 total bonds. In this case, this N also still has 4 bonds, but notice now, when it's protonated, it's going to have only 2 H's attached to it. It'll still carry that plus charge, right? But it'll only have 2 hydrogens as opposed to 3, because now it has 2 bonds to carbon as opposed to the other amino acids, which just has 1. So this one's kind of crazy, a little bit weird. This one's actually called Proline. So Proline's 3-letter code is PRO, and its 1-letter code is 1. So there's obviously fun facts about proline because it's so weird, right? So proline we say that its alpha amino group is secondary. Now, secondary just implies that the N in the amino group is bound to two carbons, right? So it's bound to this carbon, the alpha carbon, and this other carbon here that's part of the side chain. Okay, so all other amino acids, their alpha amino groups are primary because they're only bound to one carbon. So, in addition, that you can imagine that. Well, actually, excuse me. We're in the future. We'll be talking about how how these amino acids sort of connect to form long polypeptide chains and eventually proteins. Um, and they're going to be connected at amino groups and carboxyl groups in long chains. And again, we'll talk about that later. I don't want to confuse you too much. But imagine if another amino acid is connected from here to here. Right? It's another amino acid's carboxyl group is connected to this amino group on proline. Now, you can imagine the steric hindrance associated here with all this, given that the fact that the R group is attached to the alpha amino group. So there's actually no free rotation about that group, and what ends up happening is that because of steric hindrance, this amino acid, when it's implemented in proteins, it creates kinks in protein structure. Creates kinks or bends. In protein structure. And that's simply due to steric hindrance. Now, if you remember from organic chemistry, steric hindrance is just the whole idea that atoms take up some sort of space, and if they come into contact with other atoms, they're going to bump and things like that. So atoms want their space, electrons want their space. The electrons want to be sort of away from other electrons. Those like charges repel. So this clutter here around the amino group causes a steric hindrance for anything else that's going to be bound to it. So what ends up happening is that this amino acid sort of bends away because of that that steric hindrance. Okay, so proline is a little bit unusual in that sense. Okay, so again, those fun facts are things to bear in mind about proline. Okay, so far that's 7 of the 9 amino acids, of the 9 nonpolar amino acids to be specific. So let's draw the last 2. The last 2, the first one looks like this. Okay, now what I'm going to draw here is this. Now these have rings, so they're already kind of crazy. Now this one, this here, these are double bonds within the rings. This has a nitrogen here attached to a hydrogen, and the rest of these are just carbon-hydrogen bonds. This amino acid is called tryptophan. Tryptophan. And its 3-letter code is TRP. So again, that's a little bit unusual in that it's not the first 3, but it still sort of makes sense. If you saw TRP, you would probably think tryptophan. The 1-letter code for tryptophan is not actually T, it's W. And the reason why is because there's other amino acids that start with the letter T, and we'll get to that in a future video. But in tryptophan, it's actually W. Now that's kind of weird, but maybe this will help. If you look at the way this thing is drawn, if you look at it from the right, you kind of have these, if you just follow the bonds, you kind of got this shape going on over here. And that's actually a W if you tilt your head to the left. So tryptophan, if you want to remember its 1-letter code, if you remember its structure, you can kind of just, it can help you remember the 1-letter code. So things to keep in mind about tryptophan is that, now notice all these are, I've talked about all these different amino acids so far being nonpolar, and nonpolar is sort of synonymous with hydrophobic. Tryptophan, right, has this nitrogen here, so it's a little bit less hydrophobic or nonpolar than the rest of the nonpolar amino acids. It's a little bit less hydrophobic than other nonpolar amino acids. Okay, and that's because of this nitrogen here. This nitrogen is pretty electronegative, but relative to the overall structure, all these carbons and hydrogens essentially end up yielding this R group being to be nonpolar and hydrophobic, but it's a little bit less, like I said, a little bit less hydrophobic than all these other groups because it actually contains a somewhat electronegative atom. Now, another thing to keep in mind is that this ring structure here is called an indole ring. An indole structure. Okay, and I'll note that here as well. Oops, indole structure. And it's also an aromatic, has an aromatic side chain. If you remember back to organic chemistry, you often talked about benzene being the classic aromatic structure. This entire indole structure is aromatic. So this has an aromatic side chain, or R group. Okay, so that's enough about tryptophan. But again, that little W shape can help you remember the one-letter code of the amino acid tryptophan. So that was 8. That's 8 out of 9. The last one looks like this. And just to seal the deal, I might as well draw the carboxyl group, the hydrogen, and the amino group on this one, just as a reminder that all of those that I've drawn should have had those, but I wanted to save time. It's already been, 21 minutes so far. So this one looks like this. This amino acid, so it has a CH2 and then this benzene ring, right? So what this one is called is actually called phenylalanine. Phenylalanine. So its 3-letter code is PHE, but its 1-letter code is F, right? Because P has already been taken by Proline. So, but it makes sense, kind of. Phenylalanine, that has the F sound, so that shouldn't be too tough to remember. The fun fact about this one is that it's also has an aromatic side chain, right? That if you remember back to organic chemistry, a benzene rings as a substituent is usually considered a phenyl. So this is a phenyl. Well, where does the alanine come from? Well, if we scroll back up really briefly, we notice that alanine was just a methyl group. Well, phenylalanine just has a phenyl group attached to the alanine, right? This methyl group just has a phenyl attached to it. So again, that can help you remember. If you know alanine and you know what a phenyl group is, then phenylalanine shouldn't be too tough to remember. So this also has an aromatic R group as well, like tryptophan. Okay. One thing I wanted to mention was that the idea of aliphatic side chains, and let me actually scroll back up and mention that. So any structure that has just carbons and hydrogens and no rings would be considered an aliphatic side chain. So let me note that up here in red. Okay, so anything that's aliphatic simply means carbons and hydrogens, right, in the side chain with no rings. So alanine and valine would be considered to have aliphatic side chains. Okay, so that's pretty much it for the amino acids that are nonpolar. Those are their names, their structures, their 3-letter abbreviations, their 1-letter abbreviations, important characteristics for a few of them that you should bear in mind, and little tricks here and there that can help you remember them. And that's pretty much it. And keep in mind that all of these that I've drawn here are the predominant forms at a pH of 7. All of these groups, all of these R groups, are non-ionizable, which means that you can't turn them into ions, right? We'll get into in the next video, we'll talk about polar amino acids and how they sort of work. So these ones are all the non-polar ones, and I hope that was a helpful video. See you next time.